Hi, I'm Wei Zhou. In this presentation, I'm going to give a quick tutorial on a program called SAIGE, which is for scalable and accurate implementation of generalized mixed models. It was developed for conducting genetic association studies in large-scale biobanks and cohorts. We will firstly go through several challenges of GWAS in large-scale cohorts and biobanks, mostly for binary phenotypes, followed by our solutions. We have implemented SAIGE for single-variant association tests. Because the single-variant association tests are usually underpowered for rare variants, more recently, we have implemented the region or gene-based association tests called SAIGE-Gene for rare variants in the same SAIGE-R package. Last in this talk, we're going to use some examples to demonstrate how to run the SAIGE-R package. Here, three main challenges of GWAS in large-scale biobanks and cohorts are listed, including sample relatedness, large-scale data sizes, and unbalanced case control ratio of binary phenotypes. First, let's take a look at the sample relatedness. Sample relatedness is an important confounding factor in the genetic association tests that needs to be accounted for. For example, in the UK Biobank data, almost one-third of individuals have a third-degree or closer relative in a cohort. However, linear and logistic regression models assume individuals are unrelated. Therefore, instead, we use mixed models for GWAS with related samples. Mixed models use the random effect, denoted as B here, to account for sample relatedness. B follows the multivariate normal distribution, whose variance contains the genetic relationship matrix called GRM. Each off-diagonal element in GRM is corresponding to the relatedness coefficient of a sample pair, which can be estimated using the genetic data. In the linear and logistic mixed models, B, which is the random effect, is included to account for the sample relatedness through the GRM. In 2015, Liu et al. Has developed a linear mixed model method called BOLT-LMM. BOLT-LMM uses several asymptotic approaches to achieve the scalability of the mixed models, and it is the first linear mixed model method feasible for GWAS in biobank-scale data. So, can we use the linear mixed model for binary phenotypes? In this figure, the variance against the mean of residuals are plotted for the linear model and logistic model. The linear model assumes constant variance, as the orange line shows, while the true mean variance relationship for binary traits is represented by the green line. Therefore, for binary phenotypes, instead of using linear mixed model, we would like to use the logistic mixed model. In 2016, Chen et al. Developed an R package called GMAT, in which the logistic mixed model has been implemented for GWAS on binary phenotypes while accounting for sample relatedness. So we use logistic mixed models to account for sample relatedness in GWAS on binary phenotypes. When we apply the logistic mixed model as implemented in GMAT to one example phenotype, colorectal cancer in the UK Biobank, we found that it would take more than 600 gigabytes of memory and 184 CPU years to finish one GWAS in the UK Biobank. This brings out our next challenge, large-scale data. To be able to run logistic mixed models in biobank-scale data for binary phenotypes, we need to optimize the implementation. To make logistic mixed models computationally practical for large-scale datasets, we use similar approaches that have been previously used in BOLT-LMM, which is the first linear mixed model method that are scalable for large-scale biobanks. Several approaches have been used to reduce the computation memory and time. We have successfully reduced the computation memory from more than 600 gigabytes to less than 10 gigabytes, and the computation time becomes nearly linear as the sample size increases. The program optimization allows us to apply logistic mixed model for binary phenotypes in the UK biobank. Here again, we have the same example for colorectal cancer. After we apply logistic mixed model to this phenotype, we still see many spurious signals, as we can tell from the Manhattan plot. So what is missing here? We later find out the reason is the unbalanced case control ratio of binary phenotypes. Unbalanced case control ratio is widely observed in biobanks. This plot is showing the distribution of case control ratio of 1,600 binary phenotypes in the UK biobank. And around 85% of them have case control ratio lower than 1 to 100, which means there are 100 times more cases than controls. Previous studies have shown that unbalanced case control ratios can cause inflated type 1 errors of the score test, which is represented by the blue line in the plot. The y-axis is for type 1 error rates, and the x-axis is for the mean allele frequency of the testing genetic markers. The score test is widely used in GWAS because of its relatively low computation burden. As we can see, from the left to the right, as the study becomes more unbalanced, the inflation becomes more severe. Also, as the mean allele frequency becomes lower and the inflation becomes more severe. The reason is that when the case control ratio is unbalanced, the score test statistic does not follow the normal distribution. In the plot, the dotted line is for the normal distribution, and the black solid line is for the score test statistic. So when we try to get a p-value based on the normal approximation, we'll see inflation. To solve this problem, Day et al. In 2017 proposed to use the saddle point approximation to approximate the empirical distribution of the score test statistic instead of using the normal approximation. And they have implemented this as in the SPA test art package. So for our third challenge, we're going to use the SPA test to account for the unbalanced case control ratio. So in summary, SAIGE can work for GWAS with related samples based on the mixed models, and it can work for large-scale data with those optimization strategies, and it can account for unbalanced case control ratio of binary phenotypes through the saddle point approximation. Again, back to our example phenotype, colorectal cancer, and we see SAIGE successfully corrects the inflated type 1 error. We have applied SAIGE to 3 other phenotypes in the UK biobank on white British samples, from coronary artery disease, which is relatively common with the case control ratio 1 to 12, to the less prevalent disease, thyroid cancer, with case control ratio 1 to 1,000. As we can tell from the third column of the Manhattan plots, and SAIGE has well-corrected type 1 error rates. SAIGE contains 2 steps. In the first step, the non-logistic mixed model was fit with phenotype, covariates, and genotypes that were used to construct the GRM on the fly as input. In the step 2, score tests were conducted for each genetic marker with saddle point approximation applied to account for the unbalanced case control ratio of the binary phenotypes. SAIGE has been implemented as an open source R package and has been used to run GWAS for UK biobank data and for other biobanks and large consortia. PhewWeb has been created for some of the projects for browsing the phenome-wide GWAS results. As more and more sequencing data are being generated, more rare variants are being detected. However, single variant association tests by GWAS are usually underpowered for rare variants, and set-based tests can be used to increase the power, in which rare variants are grouped and tested together based on some functional units such as genes and regulatory regions. We then extended SAIGE for region or gene-based tests for rare variants. We implemented different set-based association tests, including burden, SCAD, and SCAD-O. And SAIGE also allows for incorporation of multiple minor allele frequency cutoffs and functional annotations. Here are some useful papers that you can read if you want to know more details about different methods. And the first three are SAIGE, SAIGE Gene, and the SAIGE Gene Plus, which is an improved version of the SAIGE Gene that we have recently implemented. And all of the three methods have been implemented in a SAIGE R package. And the fourth one is the GMAT, which is an R package implemented the logistic mixed model. And the BOLT-LMM is the first linear mixed model method that is scalable for biobank-scale data. And the Regini is a new method based on some machine learning approaches. It is a very computationally efficient method for biobank-scale GWAS. I would like to thank Dr. Zhangcheng Zhao, who co-developed SAIGE Gene with me, and my PhD advisors, Dr. Sean Lee and Dr. Kristen Willer, my postdoc advisors, Dr. Mark Daly and Dr. Benjamin Neal, and my colleagues from the University of Michigan and our fantastic collaborators from the Hunt study in Norway. Next, we have prepared some SAIGE and SAIGE Gene examples as wiki pages in the GitHub, and we will go through some of them together. If you click on the first link, it will bring you to this SAIGE hands-on practice. To get ready, you can install the SAIGE R package following the instructions that you may find the link here on the SAIGE GitHub. And all the example data can be found in the ext data. So SAIGE has two steps. The first one is to fit a null logistic or linear mixed model. And the second one is to perform association tests for each genetic marker that we want to test, and while applying the SPA to score tests to account for unbalanced case control ratios. Here's for step one, you can use the trait type option to specify whether it is a binary or quantitative trait. For binary trait, SAIGE will automatically fit a null logistic mixed model, and for quantitative trait, it will fit a null linear mixed model. And step one takes two input files. The first input file is in the plink format, containing genotypes of genetic markers that we want to use to estimate the genetic relationship matrix, which contains the relatedness coefficient of simple pairs. And the second file is called phenotype file, which contains non-genetic covariates, if there is any, and phenotype and sample IDs. Here's a snapshot for the phenotype file, and you can see there are multiple columns corresponding to phenotype. You want to test covariates included in the model and the sample IDs. So here's an example command line to fit the null logistic mixed model for binary phenotypes. And as we can see here, we have the plink file, and we have to specify the phenotype file, and which column is for phenotype, and which columns are for covariates, and which column is for sample ID. So there are way more options available for step 1, and for more details, and you can use call this R script and -- help and to see all of them. And then if you run the example step 1 comment in the previous slide, and you expect to see the screen output ends with the following text if the job above has been run successfully. And step 1 will generate 3 output files. As you can see, model file ending with the .rda, and the variance file, which contains a number for the variance ratio, and then association result file, which is an intermediate file. And so the first two will be used as input for step 2. In step 2, we perform single variant association test for each genetic marker. So step 2 requires 4 input files. And first one is the dosage or genotype file containing dosage or genotypes of markers we want to test. SAIGE supports 4 formats for dosages or genotypes. It is VCF, BCF, BGen file, or the SAF file. So we'll use BGen file in the example today, but you can click on the links to see more details about those different file formats. And the second one is the sample file. So this file contains 1 column for sample IDs corresponding to the sample order in the dosage file. And no header is included in the sample file. And this file is only required for BGen input, as for some BGen files, there is no sample ID information included in the BGen. But for VCF and the BCF and the SAF, sample IDs are already included in those files. So this sample file is not required for those file format. And the third and fourth input files are output from step 1. Here's an example code to run step 2. As we can see, we specify BGen file index, and sample file, and we specify the chromosome number because only 1 chromosome can be tested in each job. And we use a minimum allele frequency or minimum allele count to specify some cutoffs for markers to be tested. And GMAT model file and the variance ratio file, both of them are output by step 1. And then we use NUM line output. So we use 1000 here to tell SAIGE to output the results of every 1000 markers to the output file. We don't want to use a very low number here because it will generate heavier overhead of writing to the output file. And then the last one is output AF in case and control. If we specify this one to be true, and SAIGE will output the allele frequencies in cases and controls if we're testing a binary traits to the output. So here's an header information in the output file by step 2, which contains the association results for the genetic markers we're testing. So association results are all with regard to allele 2, as we see in the output file. And I want to point out the column called p.value.na. So these p-values are obtained under the normal approximation. So if we generate a QQ plot for p.value.na, it's very likely for us to see the inflation for binary phenotypes with unbalanced case control ratios. So the p.value are the p-values that we want to use. Next, if you are interested in conducting gene or region-based tests for rare variants, you can click on the second link here, which will take you to the wiki page we created for examples of SAIGE gene jobs. So here we can see this page contains the similar formats as SAIGE that we have went through for the hands-on practice. SAIGE gene also contains the similar step 1 and step 2 as SAIGE does. And it contains an extra step called step 0, in which we construct a sparse GRM based on the provided plink file. And this step only needs once for each data set, and it does not change according to different phenotypes. So in step 0, we're creating a sparse GRM based on the genotypes stored in the plink file as we use for step 1 for the full GRM. The difference is that we create the sparse GRM and store it in the file, and so it can be reused in step 1 for different phenotypes. So the input file will be the plink file, same as the plink file for step 1. And then the output file will be a file storing the sparse GRM and a file storing IDs of samples in the sparse GRM. So if you run an example code and you expect the screen output ends with the following text if the job has been run successfully. Then we're running step 1 again. It is to fit a null logistic or linear mix model. So step 1 asks for 4 input files instead of 2, as we see in SAIGE. So the first 2 are the same as step 1 for single variant association test in SAIGE. It's the plink file storing the genotypes and a phenotype file containing the phenotype, covariates, and sample ID information. And the last 2, the third and fourth ones, are output by step 0, which are a file storing the sparse GRM and the file storing the sample IDs in the sparse GRM. So here's an example code for step 1 job. And as we can see, we can use the 2 options that are highlighted here to specify the sparse GRM and sparse GRM sample IDs that we have obtained from step 0. Again, dash help can be used to see more detailed parameter list. And if the step 1 job has been run successfully, and the screen output will end with the following text. As we see, there are multiple values corresponding to multiple variance ratios, and they are actually for different mylittle count categories that we are using. So for the specific cutoffs for different mylittle count categories, and you can use dash help to have a look. So this step 1 will generate 4 output files, and the first 3 are the same as the step 1 output by SAIGE when we try to run single variant association tests. The model file and the variance ratio file and the intermediate file for the association tests of some randomly selected markers. And the fourth file here is specific to SAIGE gene, which is a sparse sigma file. So this file will be used as input for step 2, along with the first and the second output file, which are the model file and the variance ratio file. In step 2, we're performing the set-based association tests. It asks for 5 input files, and the first 3 are similar to SAIGE, and they are dosage or genotype file in different formats, and model file and the variance ratio file generated by step 1. Note that if you're using BGen for dosages or genotypes, and you need to use the sample file to specify the sample IDs in the BGen file. And then let's look at the fourth and fifth file. They're specific to SAIGE gene. And the fourth file is the group file. We can take a closer look at this one. So each line in the group file is for one gene or one set of variants that you want to test them together as a set. And the first element is for the gene or set name, and the rest of the line is for variant IDs included in this gene or set. For VCF or SAF input, the genetic marker IDs should be in the format chromosome, position, ref, alternate allele. And for BGen file, the genetic marker IDs should match the IDs in a BGen file. And each element in the line is separated by tab. And the fifth file is called sparse sigma file. It is also output by the step 1. With all of those input files, and we can run step 2 job to perform set-based association test. And I want to point out this option called max MAF for group test. And this can be used to specify the maximum allele frequency of genetic markers that you want to include in a group test. So by default, this value is 1%, but if you want to test rare variants only, and you can lower this number. Again, uses -- help to see more detailed parameter list and information. And if your step 2 job runs successfully and you see the screen output ends with the text as we show here. Then let's take a look at the output files by step 2. Step 2 will provide a file with region or gene-based association test results. For each gene or region we're testing, we'll provide p-value of SCAD-O test and also p-value from burden and SCAD test. And there are a couple of columns there showing the number of markers falling in each allele count category. And the category information can be found in the step 1 script. And if we specify that we want to output the single variant association test result for genetic markers in the genes or regions we're testing, and the second file will be generated to provide those information. Last, if we want to use multiple allele frequency cutoffs and multiple functional annotations to test each gene, for example, and we can use different group files and to run step 2 for that gene for multiple times. So to combine those different p-values for the same gene, and we can use the Gaussian distribution to combine them. So here's the code that you can use to combine multiple p-values for gene or a testing region.